Howdy folks, welcome to the home office. I'm going to try to show you how to do the genetically sketch artist lab. Let me swing around my overhead camera here for a second. Hang on. Here we go, overhead camera in place. This of course is going to work much better if you have your own copy of the genetically sketch artist lab. Let's make sure my pen works here. So make sure you print one of those up or have one. You're, of course, going to read through all this. I'll try to hit the highlights. I know some of you have already started this in class. If that's the case, just wait till we catch up. And even so, I might show you a faster way to do this. So the idea, of course, is that we're trying to track down this individual who's left behind some DNA evidence, Smelly Jim Sock. And strangely enough, it's a single strand of DNA, which right away, you know, that doesn't really work. DNA exists double-stranded, but I've given you what's called the coding strand. You need to create the template strand. It's part of the exercise, so you can get used to, you know, A's and T's and G's and C's, or A's and U's and G's and C's, you know, whether you're doing DNA or RNA. Anyway, in the instructions here, they give you all the pertinent information. Like what we're trying to do is identify those sections of this DNA strand that actually code for amino acid sequences that impart traits. And then from those traits, we're going to, and here they are, here's the amino acid sequences, and they code for particular traits. And from these traits, we're going to get a picture of the person who left the smelly gym sock. So we have to be able to find them. All right, now I put together this list here. Let's switch over to the list, to this transcription sheet of the same, this is the same strand of DNA, right? That's over here. All right. And what we should do, of course, is go through and everywhere there's an A, you're going to put a T. Everywhere there's T, there's an A, T, A, G, C, C. All right, I'm going to stop there. Okay. Now, hopefully, some of you guys have probably already done this for homework. And then, of course, you would take the complement here, which is the template strand, and we would convert that into a messenger RNA transcript, right? And of course, T goes to A goes to U. It would go to T if it was DNA, but it's RNA, so it's a U instead. And that's the pattern is the same, and it just goes down. Okay, so now our task, of course, is to actually find the gene. Now, you may have noticed that really all we've done by going from A to T and then back T to A, what I've really done here is we've essentially copied over this coding strand into messenger RNA, except wherever there's a T, we put a U, right? So these are actually the same. Now that's going to help us, right? Because what we have to find is we have to find the part of the DNA that actually codes for the gene. Now, the gene's going to start out with a start codon. The start codon is AUG. All right. And the gene is going to progress all the way down, codon by codon, right? Those three bases, all the way down until you get to a stop codon. There are three stop codons, UAA, UAG, and UGA. They're all written down there, so you can refer to those. All right. So all we got to do is we have to find our first start codon. And here it is right here, AUG. Start codon. And then we're going to need to translate each one of these codons until we get to the next stop codon. Now I know AUG is a start codon. It's okay. It turns out AUG actually codes for methionine. So we can put a met right there. Or it's actually an amino acid. All right. And just because we've already hit another start codon, we've actually already started. So it's okay if you walk into a room and the lights are already on. Do you turn on the lights? No, they're already on. You don't have to. So we've already started. We know that codes for methionine. So we just keep going until we get to a stop codon. All right. And here is UAA. Here is our first stop codon. And so there's our first gene. Between this methionine and this stop codon is our first gene. Now, what we would need to do then is go on down the line until we find the next start codon. Now, we don't have to actually make the messenger RNA transcript because remember, this is the same thing. So all we really have to do is find an ATG because that would be our next start codon. And here's ATG. So that would be AUG, which we know codes for methionine. And then we have to do ACU, AGU, UGG, CCC, GA. UGA is one of our stop codons. All right, so we're going to have to translate each one of these genes. Okay, so how do we do that? Well, let's see. AUG. What we're going to need is our translator table. Now, you can use the one that's in the lab, or you can look one up online. They're all the same. All right. Remember, every organism uses the same one. So we have to find AUG. All right. And the way we do that, the first one over here is tells us which row it's in. Right. And it's A. So we know it's in this row. The second one, nucleotide is across the top. Right, the second base. So it's in this column and this row. So it's in this box. Which one is it? Well, that's this row over here. G is the bottom one, methionine. So that's why we put met there. Next one is GUA. So GU. Oh, look, it doesn't matter what the third codon is. They're all valine. So valine. GC. Oh, again, doesn't matter what the third one. Alanine. Okay, next one. UUA. UUA is leucine. That one we got to be a little careful of. LEU. GC. Well, we got to make sure we get this. GCG. Good. Okay, almost messed that one up. GC. Again, doesn't matter. Alanine. We already got AUG as methionine. AUU. AUU. ILE, isoleucine. So there's our first gene, our first polypeptide. We've transcribed the gene, right? Here's the gene. We've made our messenger RNA transcript. And now we've translated our first polypeptide. Now the trick is just match this up. Met, val, allelu, alamet, isoleu. Right, with which one of these? All right, let's see, they all start with met. Here's the vowels. Met, val, ala, so it's between these two. LEU, ala, met, isoleucine, boom, 11. So we know that this character has small beady eyes and poor vision. Now the trick is translate this gene, and then find the next codon. Oh, look, ATG. That's going to be AUG. Start. Go. Now, one other thing that you're going to notice, all of my genes are the same length. 1, 2, 3, 4, 5, 6, 7. So you're going to have the start codon, and then 7 more, right? Because they're going to be 8 more. All right, keep going. Find those start codons, transcribe, translate, draw me a picture, answer the questions. Have a good day.